Welcome everyone to our third webinar in our currently ongoing series. My name is Marie Latulip and I manage some of the nutrition programs at ILSI North America. This series is brought to you in collaboration with the American Society for Nutrition, and what we're doing is we're exhibiting the ongoing work of both faculty and students at the University of Illinois, and all of this work focuses on the gut-brain axis and the gut microbiome. So just to remind you that everyone is going to be on mute, but as we go through the webinar, please feel free to type your questions anytime in the chat box, and we will start going through those at the end of the session. So quick note on ILSI North America, for any of you that might not be familiar, we are a nonprofit scientific organization, and we focus on advancing nutrition and food safety science. And we do not lobby, we always like to make that clear. Intellectual contributions of scientists from these three sectors, government, industry, and academia, are combined with pooled funding, and that allows us to advance the science. So we just wanted to thank everyone for joining today. Our audience reflects the tripartite approach that we apply to all of our activities. And if you would like to learn any more about our programs, please feel free to send us an email. You can see my email, and Gwendolyn Jones, the executive director, her email is there. And you can also visit our website at lcna.org. Also, feel free to link with us on LinkedIn and on Twitter as well. So with that, I am very pleased to introduce our first speaker, Dr. Janina Pepino. Dr. Pepino is an assistant professor in ingestive behavior in the Department of Food Science and Human Nutrition at the Division of Nutritional Sciences at the University of Illinois. So we would like to welcome her today to tell us a little bit more about her work on low-calorie sweeteners. So Dr. Pepino, we are turning the platform over to you.
Great. Okay, so thanks, Marie. And I would like to first thanks to this ILSI North America and the Society, ASN, and in particular Dr. Elvira de Mejia, the director of our Division of Nutritional Sciences, for inviting us to be part of this. We really are very excited. It's a fantastic sessions of interesting webinars, and we're really honored with this invitation. So thank you so much for this opportunity. Moving on into things, I also want to make sure that I acknowledge my lab and my collaborators. I have the luck and the pleasure to work with a fantastic group of people. In particularly, I like to thank to Clara Salame, who you will hear in a few minutes. And since she has no camera, I can tell you this is Clara here. And also Clara, as you will hear, she's working on this project on artificial sweeteners, in particular sucralose, on how it may affect glucose metabolism. And also Alex Nichols, who was my first master student in the lab and also participated in some of the studies I will be discussing with you today. Of course, my collaborators at Washington University in St. Louis and our funding agencies. I have no conflict of interest in this line of research. All the studies I will be presenting today have been funded by pilot and feasibility grant from NIH, and we are currently funding our sucralose study for the American Diabetes Association, by the American Diabetes Association. So moving into our topic today, low-calorie sweeteners is a very controversial topic, and it brings a lot of visceral responses. I wanted to start by really bringing you a very robust fact to make you remember, if you already know, or make you aware that attraction to sweet taste is universal and is innate. As you can see here in the slide, depending on what part of the world, maybe is enjoying chewing of a raw sugar cane or refined sugar or candies. What they all have in common here is all children are here enjoying the pleasurable sweet taste. We are hardwired. We're born with this hardwired attraction to sweetness, and it's believed that it helps us well evolutionary because it attracts us mammals to our first food, mother's milk, and fruits. In part, the way that it attracts us so much is because it engages important neurochemicals that make us feel good, and it's very well established, for example, in babies, that there is a very strong morphine-like effect of sweetness. And as you can see here, for example, scientists have capitalized on babies going through some painful episodes, such as when they get the first blood draw, within a few hours they're born. And you can see that in the y-axis is the percent crying is significantly reduced when babies are given a little bit of sugar water to taste compared to if they only get water. And here the dependent variable is percent crying, but this has been very well studied, and you obtain the very same effect if you look at, for example, heart rates or EEG of the brain, or if you look at facial reactivity. Really, there is a blunting of pain by the sweet taste. And we know it's the sweet taste, and it's not the calories of the sugar or the post-absorptive effect of sugars, because you can find exactly the same effect if you use aspartame, one of the low-calorie sweeteners. So we know that there is an effect, there is something beyond calories, and I will be talking about this topic, will be recurrent in my talk. Beyond calories, we know sweet taste has powerful physiological effects, and early on in life is a very good analgesic. So we really like it. It makes us feel good. It is associated with the release of endorphins, with dopamine. How much we like it? I would say we maybe like it too much for the environment that we have to live on today. As you can see here, this is one of our last publications, but it has been shown for many years in the 70s. And we know that in particular, some groups are more vulnerable to this very huge attraction to sweetness, such as children and adolescents that really like a lot of sugar compared with adults, for example. And you can see here our most preferred level of sugar in water. And you can easily see here that the regular sodas are tuned to the palate of adults. If it was for children or adolescents, we like even more. Here, each of these cubes is like 4 grams of sugar. And you can understand why it's so difficult for us and why more than 70% of Americans are consuming more than the recommended and what would be considered the maximum amount of added sugars that many health agencies are promoting, which in case of children or women is no more than 6 or 7 sugar cubes. So why is it that why is it that we need to reduce the consumption of something that makes us feel so good and we're attractive since we are born? Well, this is not news for us. We know now for many years now has been shown this association, and now by the collection of evidence, we can say that is a robust evidence that the consumption of added sugars is associated with disease, with metabolic problems, with obesity, with type 2 diabetes. And therefore, what I did here is just to illustrate the point. I just look at one of the most robust recent meta-analyses has been shown. In this case, they look at 17 prospective cohorts in which they look at the association between the consumption, in particular, of sugar-sweetened beverages and the risk of developing type 2 diabetes. And you can see that overall in these 17 prospective cohort studies, there is an increase for every 1 serving of a sugar-sweetened beverage. There is an increase of 18% in the risk to develop type 2 diabetes. But you just heard I said also that the consumption of added sugar is associated with an increased risk to develop obesity. And we know that obesity is very comorbid with type 2 diabetes. So when they control for adiposity, even after they control for adiposity, you can still see that there is an increased risk, in this case reduced. So some of the effect that we saw, that increase of 18% is reduced to 13% for each serving of sugar-sweetened beverage to the risk to develop type 2 diabetes. But you can see that even independently from adiposity, the consumption of added sugar is associated with diabetes. And therefore, it's not surprising that many governmental as well as non-governmental health agencies are really following the idea of reducing the amount of added sugar in our diet. The USDA now in the 2020 dietary guidelines, they moved from 10% that it was in 2015. Now they're recommending less than 6% energy of added sugars, which really goes very close to the less than 25 grams per day in children or women that recommends the American Heart Association. So, okay, on one side we have the biology that make us vulnerable and really enjoy and feel pressure for sweetness. On the other side, we have the metabolic problems and how unhealthy it can be having this added sugar. So one way that maybe we can solve this control syndrome would be getting to consume more of these low-calorie sweeteners, which is providing us with the pleasure of the sweet taste with few, if any, calories. And there are many that are in the market. Most of my talk will be really talking about sucralose. And the way they work is, as you can see in this cartoon here on the left, the way that we perceive sweetness is when specific molecules, and these are very different, diverse chemistry, they interact with the sweet taste receptor localized in specific taste cells in the taste buds in the mouth. And you can see that this sweet taste receptor, as illustrated in this cartoon, is a dimer. The T1R2 and T1R3 units, and by having this dimer, you can see there are many opportunities of different binding sites, so many different chemicals, very different structures can activate it. And once these ligands interact with the sweet taste receptor, they will, this is a receptor that is coupled to a G protein that is called alpha-gas-leucine, and this protein will engage in a cascade of events that ultimately will give us to this sensation of sweetness. Now, as you can see here, the amount of the sweetness imparted by some of these different molecules is very different. And these low-calorie sweetness, what happens is that they're so efficient in activating, so intensely activate the receptor, that you can see that, for example, compared with the relative sweetness of sucrose, if you put that at 1, sucralose is like 600 times more sweeter, and therefore it requires very little of this compound in order to provide the level of sweetness that you need, for example, to sweeten something with sucrose. And that's why, therefore, not much calories. And so it sounds like an ideal situation where you have the sweet taste and no calories. However, there is a lot of data, a lot of controversy over the fact of many of these mostly observational data, epidemiological data, but I hope I can convince you today that there are also other side of data that we need to start paying attention to and understanding what can be explained from this kind of controversial data that creates so much debate. There is this evidence, although limited compared to the evidence that we have for other sugars, but there is also this evidence that the consumption of sugar substitutes increases the risk of type 2 diabetes. So to put it in perspective, this is the same meta-analysis that I shall show you for added sugar. This is looking into artificial sweetened beverages, and you can see that overall, they also found like a 25% increase in the risk to develop type 2 diabetes for every 1 serving of an artificial sweetened beverage consumed. Again, if you look at what happened when they control for adiposity, they see that the association gets diminished. And in the case of the low-calorie sweeteners, it is decreased much significantly than the one that we saw for added sugar. So now instead of a 25% increased risk, there is an 8% increased risk when they look for the association between the consumption of low-calorie beverages artificially sweetened and the risk of developing type 2 diabetes. So this is kind of generates some controversy. Some people say that all is going on here is a reverse causation. So when we try to understand this association, it's several in the literature, it is in many studies, the association between low-calorie sweetener consumption and increased risk for different metabolic diseases, including the risk of type 2 diabetes. The first potential mechanism that comes to mind, and is in all the discussions, is the idea of a reverse causation. And it may be very well that, like this kid that is sitting here in the plane, and he thinks that every time that the pilot turns on the light that he has to wear the seatbelt is the pilot causing the turbulence. It may be very well that people on a track of gaining weight or with a heavy family history of diabetes, that they choose to consume these products in order to try to avoid what they have, like a heavy charge by genetics or the environment to develop type 2 diabetes. So in that case, this low-calorie sweeteners association is because they are more of a marker of other things that have to do with being already having obesity or having a high risk to develop diabetes than actually a cause. Another very common thing that can be happening, and here I would like to make the note that none of the mechanisms I will be talking today are mutually exclusive, so they can all be happening at the same time. We are really bad at calculating calories and particularly good at miscalculating, so we many times think that we are saving so much calories for our diet soda that we can engage into having an extra layer of cheese or dessert with the lunch, because we are going to make up equal, and we ended up actually eating more calories than we anticipate. But I would like to move our conversation beyond calories and beyond eating too much and compensating from the sugar. I would like to open our brain and explore other possibilities. Is there any chance that these low-calorie sweeteners could be bioactive, that they could engage in mechanisms that has something to do with the metabolism of glucose that make us more sensitive to develop type 2 diabetes? And among the different potential mechanisms, of course, one that comes to mind that has been in the news lately a lot, and that is very pertinent for our topic for this series of seminars, is the data showing that low-calorie sweeteners can interfere with the intestinal microbiota and induce glucose intolerance. Although this is very central to the seminars, topic of these seminars, it's not my area of expertise, the microbiota. So I will just mention this as a fact that there is a lot of data. Most of it, the most robust data, comes from preclinical models, studies in rodents, showing how low-calorie sweeteners, in particular, there is data with sucralose, with saccharin, but there is also some data with others. They can interfere with the microbiota and induce glucose intolerance. They can transfer the phenotype of glucose intolerance from animals that had never had exposure to a low-calorie sweetener just by transfecting with the feces of an animal that had a diet heavy consumption of the low-calorie sweeteners, and they show this phenotype of an induce of a glucose intolerance. In people, the data is at best preliminary. There's studies with small sample of people, but they are very convergent with the data that shows in preclinical models that there is something going on between the interaction between these sweeteners and the microbiota. The mechanism that I would like to talk more about today, that is also related with our seminar, which is related with the gut-brain axis, is to try to get into some of the most innovative findings in this area of the gut-brain sensory transduction that happened over the last few years. And they start to show this pattern that the low-calorie sweeteners can interact with sweet taste receptors that are located in the gut, and that these receptors are playing a role in glucose absorption, for example. So this, to me, was one of the most exciting findings when I started deciding to look at the effect of sucralose in glucose metabolism. I am a sensory scientist for training, and then is when I start really getting involved more on what happened with glucose metabolism. When they found that the taste receptors weren't just for the tongue, and that they are, it looks logical now, that they are expressed in many tissues in the body. And we know that sweeteners, both caloric and non-caloric, can activate sweet taste receptors that are localized in different tissues, including the intestine and the pancreas. And in this cartoon, they show that just as I was describing for what happened, the situation in the mouth and how the T1R receptor, the heterodimers, coupled with the G protein, is expressed in the taste buds, they found very similar arrangements in the gut, in the intestine, and in the pancreas. And you can see in the intestine, the sweet taste receptors, by activation of G proteins, are engaged in the expression of sodium glucose transporters and contributing to the absorption of glucose in the intestine. They are also related with the release of the incretins and helping in the amplification of insulin effects. In the pancreas, the expression of these receptors, we still don't know precisely all the mechanisms that it's going, but all the secondary messengers and all the cascade, but there is data very robust in preclinical models that show that these sweet taste receptors are also engaged in the release of insulin. Now, remember when I talk here about in the intestine, the expression of the sweet taste receptors in the intestine and how they collaborate with the expression of the sodium glucose transporter 1, because I would say that this is another really exciting finding that comes from the lab from Bohorquez in Duke, where they really start showing a more emerging biology of a new gut-brain sensory transduction. So what we knew before this data from the lab, before that finding of what they coined the neuropod cells, we knew of these enteroendocrine cells in the intestine expressing these sweet taste receptors and other receptors for nutrients. And they call, as you can tell by the name, enteroendocrine, so very biased to the only way of communicating we knew from the gut to the brain through the vagus was by the activation, the release of hormones. and so therefore these enter endocrine cells. But now we know that the sensory transaction by these cells, epithelial cells in the intestine, is not unique, is not exclusive by hormones, but they discovered this beautiful arrangement by which these cells that they call the neuropod cells, they have co-localized hormones as well as some neurotransmitters, and they can connect to the vagus via synapses. So the gut in milliseconds can know if they are sensing what they're sensing, that they have glucose in the intestine. In milliseconds, they will know. So this is a gut-to-brain relationship, a gut-to-brain connection that now got really bumped up, that is not just so slow and through hormones, but it also can happen in a question of milliseconds by a synapses. And actually, this group has a paper that is now in medical archives. It's a preprint that it shows that not only sucrose or glucose and other sugars can activate the neurotransmission in this neuropod, but also sucralose can activate and cause a vagal response via this neuropod signaling. So we know that diets that are high in low-calorie sweeteners, we knew this from diets that were high in sugar in animal models, but it was shown that diet high in these low-calorie sweeteners can upregulate the expression of sodium-dependent glucose transporter. And as you can see in here, in this series of results, where you are expressed in the y-axis is the expression of sodium glucose transporter. And when you have in black is animals that were exposed to a low-carb diet, the regular chow. And in orange is the same regular low-carb diet that is spiked with sucralose. And after a couple of weeks of consuming this diet full of sucralose, well, spiked with sucralose, what they found is that there is increased expression of sodium glucose transporters in the intestine, which is functionally effective in increasing the rate at which glucose can be uptake in the intestine. And as you can see here, if you look at this same exposure of the diet in animals who are knocked out, either in one of the parts of the sweet taste receptor, the T1R3 dimer, or the alpha-gas-leucine, you can see that this increase in SGLT1 is totally blunted. So the increase in SGLT1 expression in the intestine that contributes to this increased uptake of glucose in the intestine at the velocity that you can get the glucose uptake is mediated by the sweet taste receptor. And I know it is very exciting when we find these very nicely and elegant studies in rodents, but we also think, well, maybe there are species differences. I would like to point out that a very similar phenomena has been shown in pigs, in cow, and now more recently by the same group in rabbits. So here we have 4 different mammal species in which the exposure to low-calorie sweeteners can cause a physiological effect associated with this increase in the expression of the sweet taste receptors that increase the sodium glucose transporter. In rodents, I show it with sucralose. The sweetener used for the studies in piglet is sucran, which contains saccharin, and in rabbits with stevia leaf. So with this background, one of our first questions in the lab was to understand, okay, we know it is happening in animals, it's having an effect, it's biologically active. Can we see any difference in people, metabolism of glucose? So I was at the time working in Washington University in the Center for Human Nutrition. And so I had really good support and really great people to work on glucose metabolism. And we decided to study people with obesity. I wanted to use my first pilot and feasibility money in a group that I thought was the most clinically relevant, because people with obesity are the ones who are more likely to consume. They are the ones who more consume low-calorie sweeteners, and the ones who receive the advice to consume them more frequently. And so we look at people with obesity, but we want to make sure they were non-regular users of low-calorie sweeteners coming from the sensory field. I'm looking at all the data that we knew at the time. We only knew at the time what happened I think, in the piglets and in rodents, but we knew that diets higher in sucralose could somehow modified already the rate that glucose was going to be absorbed. So I looked for the most naive population that I could. I know they probably were exposed somehow here or there to some low-calorie sweeteners, but they were not choosing to consume this every day. They came in the lab on 2 visits, so everybody's going to be their own control. And we use a very simple paradigm in which they drink at time zero a glucose tolerant test, 75 grams of glucose. We follow them over several minutes, exploring what happened with their responses, glucose responses, insulin responses, and other hormones. And the only intervention that we did in these 2 visits is that 10 minutes before consuming the glucose, we give them to drink a glass of sucralose, a small cup with sucralose, containing sucralose. The amount of sucralose that we use is the one that you will find in one diet soda if it was all sweetened with sucralose. So it's a small amount, like a very small amount of sucralose. It was 48 milligrams. On the other visit, they received the same volume, but just water. So the idea here is that if sucralose is inert and it was doing nothing, then it will be very similar, the pattern that we'll find on glucose and insulin, whether you drink water or sucralose 10 minutes before having your glucose test. And the order of having drink glucose or sucralose was counterbalanced. So when we look at our results, we found that actually they were not identical or very close by what happened on the 2 days. And to guide you in how we're going to be reading these graphs, you can see that on black is the day that they drink the water, the control day, 10 minutes before drinking glucose. On orange is what they drink the sucralose. And you can see here in the left is the graph for glucose, in the right is for insulin. And you can see that sucralose changed, alter the glucose and the insulin time concentration curves in these participants with obesity. It increased the peak, the peak of glucose in plasma, and it really significantly increased the amount of insulin, the concentration of insulin during the OGTT. If you look at the area under the curve, there was an increase of around 20% insulin in the area up under the curve. So we thought it was very interesting. We wanted to continue moving forward. I wanted to do some more studies, and when I write my first grant to look to the NIDDK, it was like, there was a lot of excitement, but at the same time, I was asked many times this very simple question, like, why do you find this effect in your hands with sucralose, changing the OGTT, when there are many other labs, and you were not the only ones looking at that time on what happened with an acute effect of sucralose on an OGTT. But as you can see here, many other groups were looking exactly at the same, and no matter if they were giving them intraorally, intragastric, intraduodenally, you can see that all they found were no effects. And then I started saying, well, but we really study only people with obesity. And so maybe there is something different in people with obesity in the metabolism of glucose and how they respond to sucralose in those with obesity from those who don't have obesity. And I tried to convince by putting this table and said, when there is only lean, there were no effects. When you start putting some obesity in the picture, then you start finding some metabolic effects. Another thing that we don't know is that in our hands, I was coming from the sensory field, and I really wanted to have people who were not regular users, but very few people control. In this case, all the studies until that time, none of them controlled for non-nutrient sweetener use when they were recruiting from the population. So that brings us to the second study. And the way we design it is, okay, we want to determine the effect of sucralose on the metabolic response to an OGTT, both in people with and without obesity. Let's look at the data. We also wanted to first, our specific aim was we wanted to look for replicability. Can we see the same thing, response in insulin that we saw in those who have obesity? Are our studies generalizable? If you look to normal weight participant when you control for the low calorie sweetener use? And then this was a very interesting question that I got from one of the reviewers, so I'm very thankful, maybe sitting in the audience. I thought it was really good. Because the question is like, well, this increase in insulin that you see and this difference in the increased glucose that you find, the increased peak in glucose, do you see it? How do you know it's a post-oral effect? So it's something that is happening when they swallow the sucralose and, all the enticing story that you're telling us about the sweet taste receptor beyond the tongue. But how do you know it's not something mediated cephalically through perceiving the sweet taste that affect the way that then they process the glucose? And I thought it was a really good question. And so the way to look now, to control, to dissect this effect in our study, when they consume sucralose, they have both. They have the sweet taste, the cephalic response potentially, and they are consuming it, so the post-oral effects. So we will include in this new study a new condition in which people will just taste the sweet taste of sucralose, put the same amount of sucralose on the same medicine cup that they take the day that they drink it, but then they spit it out, and then they would drink 10 minutes later the OGTT. So this is what we call by sucralose sham fed. So in our second study, it looks very much to the one that I already explained to you. The only difference is that now we're going to have two groups. We wanted to compare people with obesity versus those of normal weight, and we add a new condition. In addition to drinking water or sucralose 10 minutes before the OGTT, as I just mentioned, one day they will taste the sucralose, spit it out, and then 10 minutes later drink the glucose tolerance. The study was more complicated than that because we also used tracers, trying to understand the fate of the glucose that they drink versus the fate, what happened with the glucose that is endogenous produced by the liver, or what happened with. So if you want, you can see the study, and I'll be happy to talk offline. We looked two different tracers. We didn't see many effects of sucralose on that, and so in an interest of time, I will not present it here. So what we found in terms of comparing to our first study and now including this new group of normal weight individuals, we found that sucralose consumption modestly increased in a small amount glycemic responses similarly in both groups. And then the sham fed kind of fall in between and didn't do much different. As you had before, water and the control day when they drink water 10 minutes before glucose is in black, sucralose 10 minutes before the OGTT is in orange, and now the new group when they sham fed sucralose will be in blue. So overall, there was a main effect of consuming sucralose. It was the same for both groups, and there was an increase in glucose area under the curve of around 30% for both groups on sucralose compared to water, but not with the sham feeding. Now, when we look at what happened with the insulin responses, as you can see on the left, we replicate the effect of people with obesity. We see again that there was an increase in the amount of insulin compared to the water day, although it was of a smaller magnitude than we saw before. Before we saw a 20% increase. This is a whole new batch of new people, and then we found a 16% increase. But the most interesting finding is that there is an interaction, that where we find again this increased insulin in people with obesity. In those of normal weight, there was a decrease in the insulin concentration on the first 40 minutes after they drink the OGTT. And even the more interesting part is that when we look at the sucralose sham feeding, so just tasting something sweet, the sucralose, and spitting it out, it has very similar effects in the insulin concentrations in those of normal weight, and it's a reduction in how much insulin there was on board compared to the day that they just have water before the OGTT. And in people with obesity, as you can see, like in those of normal weight, just tasting the sucralose also caused at the very beginning a reduction in insulin responses. So I will argue that the difference here between orange and blue, in both times they taste the sucralose, but in orange they also swallow it. That is a difference in the post-oral effect of sucralose that we don't see in people of normal weight here. But it's interesting to see that there is some effect of just tasting something sweet that was similar in both groups of these non-regular users of artificial sweeteners. I want to show you here a little bit more data because I know 16%, 20% insulin area under the curve it doesn't look like a lot. But I want to remind you first that the dose that we're using is like what would be in one diet soda if it was all sweetened with sucralose. I want to show you that it's a very small effect that is very consistent. So here what you see, this is the average in insulin area under the curve on the day of water and on the day of sucralose. What you have illustrated here is for each person, each of these letters is one individual. And if you look at their area under the curve of insulin on the day they drink water compared on the day they have sucralose, you can see that in most of the 9 of 11 subjects, it was more insulin the day that they consume sucralose before the OGTT than on the day they consume water. And it's very similar also to what we found in our previous study. It was also the case that 11 out of the 17 subjects showed this response. So in summary, for the study that I should show you, sucralose taste before an OGTT decreased the plasma insulin an hour later. And this make us puzzle and start thinking on what could be the physiological role of the sweet taste in handling the glucose, a metabolic response to glucose. We also found that the ingestion, so now moving from the taste to the ingestion of sucralose had a different effect in postprandial glucose metabolism in our subjects with obesity from those of normal weight. Now, keep in mind, these are all non-regular users of low-calorie sweeteners. we found that they have similar effects on increasing the area under the curve of glucose. Whoops. However, they have opposite effects on plasma insulin concentrations between the groups. So in conclusion, when we look at the metabolic effect of sucralose, for our first specific aim, I would say yes, it was somehow replicable in subjects with obesity. The effect on insulin was very modest, but it was consistent. Can we generalize it to normal weight subjects? Our study told us that no, even after you control for the low-calorie sweetener use, it was not generalizable, but it may help us explain some discrepant results in the literature. When we look at potential metabolic effect of sucralose, perhaps we need to look at separately what happened with people of normal weight and what happened with people with obesity. Same in studies with in epidemiological studies. Maybe instead of controlling for the effect of obesity as if it was a confounder of the effect of sucralose, we should also consider the interaction in that perhaps the effect of some low-calorie sweeteners, in our particular case, sucralose, may be different in people with obesity than in a metabolic with obesity than in a person with the metabolism of a normal weight individual. So in conclusion, sucralose may have adverse effects on glucose metabolism in people with obesity, which is the group that most frequently consumes and receives the advice to use the low-calorie sweeteners to facilitate weight management. Of course, there is a huge however. The study is limited in the sense that these are clinical studies, the sample sizes are small, but they're typical from our type of tracer studies or metabolic studies. But we want to see more studies reproducing what we are showing. And it has some limitations because you know this is an acute effect of sucralose. It's what happened on one single event that they drink sucralose before having an OGTT. We want to understand what are the chronic effects if you consume them. What happens if you evaluate people who are habitual users or non-habitual users of the regular of the low-calorie sweeteners. We may want to study not just people with obesity or without obesity. What about insulin sensitive versus insulin resistant, people with type 2 diabetes? These are all studies that conducted in adults, what happened with children. And of course, we cannot consider low-calorie sweeteners as if it was one thing. We want to know also what happened with others. So far, what I show you is with sucralose. But in conclusion, my take-home message from my part is that I want to bring back the story that, you know, we know that for sugars, there are effects that are more than calories. We know that there are physiological effects of sweetness. And therefore, I think that we need to move our talk, our discussion on whether the low-calorie sweeteners are helping us so much in calories or no calories, not contributing to the diet and obesity. We need to think that maybe some physiological effect of sweetness that are related with the effect of sweetness in glucose control. And Clara will talk a lot about that and how we are handling that line of thought in her talk. For now, I hope I help you understand why I say let's use sweetness as a treat. So that is my last slide. I don't know if we have time for some questions or if we will move directly to Clara.
Hi, Dr. Pepino. Let's, I think we have time for at least one question in between. So one question is, when you talked about the neural response to low-calorie sweeteners, so linking with the receptor and then going through the gut-brain axis, is there any information about the magnitude of that response for sweeteners versus sugars that contain energy?
So if I understood correctly, I'm just thinking on the data that I showed from the lab, from the neuropod. And these are all studies in animal models. And so this is in rodents, and they're measuring the vagal response, right? If I recall correctly from the graphs, the magnitude of the responses that they found, I think they're very similar. When they look at the different sugars, the only one that didn't produce a response was fructose, for example, glucose, sucrose, and many of other, like I think also maltodextrin, they were all showing a response. Fructose did not. Sucralose, aspartame, and many other sweeteners also show a response, and it's of the same magnitude on the scale that they're measuring for the recording from the vagos. What they do, and this is in a preprint, so we need to wait until this is published, but they show also very interesting that, for example, sucrose release a different neurotransmitter is connected through the vagos by different chemistry, like the sucrose release glutamate versus sucralose increased ATP, for example. So how the brain incorporates these differences, perhaps it's one way of understanding how the brain so quickly can tell if even if your brain, even when you're tasting two things can be equi-sweet and you don't distinguish them immediately in an acute phase which one is which, your brain and fMRI will know and will activate different areas to tell you if it comes with or without calories. So now we know that it's perhaps by this communication with the neuropod that in milliseconds the gut can tell the brain if it was sucrose or sucralose.
Okay, great, thank you. I think in the interest of giving Clara plenty of time, we will transfer over to her. So our next speaker is Clara Salome, and Clara is a PhD student in the Division of Nutritional Sciences. Clara is also a registered dietitian who has a master's degree in clinical and public health nutrition. So with that, we will turn the platform over to you, Clara.
Hi, can you hear me well?
Yes.
Great. So I'll share my slides. Okay. Everybody's seeing the slides.
Yep, looks great.
Okay, perfect. So we will continue the talk on low-calorie sweeteners and if it's a trick or a treat. I would like first to thank ILSI and the Department of Nutrition Sciences at the UIC, as well as the American Society of Nutrition for giving us the opportunity to talk today. So following up on what we just heard about the gut-brain axis from Dr. Pepino and how sweet taste signaling is taking place in the gut and how it may play a role in glucose homeostasis, we will continue this talk on focusing this time on the opposite direction. So that is how the brain-gut axis. So we would like to understand what is the effect of sweet taste through the cephalic response and through that pathway, the glucose metabolism. So our data suggests that there is a physiological role of sweetness. If you recall, there is a decrease in the amount of insulin concentration whenever sucralose was tasted just before swallowing an OGTT. So here we would like to focus on the blue line, which represent tasting sucralose 10 minutes before an oral glucose tolerance test, and then spitting it out. So we noticed that compared to tasting water, tasting sucralose 10 minutes before the oral glucose tolerance test resulted in a decrease in insulin concentration in both people with normal weight and with obesity. So just tasting sucralose before the oral glucose tolerance test had an effect on glucose metabolism. On the contrary, the group of Curiasis showed that if you block the sweet taste with lactisol, which is an inhibitor of the sweet taste receptors, on an oral glucose tolerance test, we see the opposite, so an increase in insulin concentration. So to understand more here, in this group, participants were given a regular oral glucose tolerance test with glucose. They had to drink 75 grams of glucose at time 0, which is here. And on the intervention day, which is the gray line, so it's the day where they were given lactisol with the glucose. And just as a reminder, lactisol is the sweet taste inhibitor. So when this was inhibited, no sweetness was detected, and we had an increase in insulin concentration that was observed. And the results also suggest that sweet taste receptors may have an effect on glucose metabolism. I would also like to point out that in this study, lactisol is blocking the receptors in the mouth, but also potentially the sweet taste receptors in the gut. So this data also suggests that sweet taste receptor signaling has an effect in insulin responses. Taken together, the studies suggest that when there is a sweetness detected before the OGTT, insulin concentrations goes down, and when there is lack of sweet taste signaling, insulin goes up. So they are kind of complementary. And this suggests that there is a physiological role of sweetness in regulating the insulin response to a glucose load, and that sweet taste receptors may have an oral and a post-oral effect on glucose metabolism. So what other evidence do we have on the importance of sweet taste and the regulation of physiological response? So there are a lot of data from the preclinical models from the group of Sweeteners. showing that sweet taste plays an important role in the prediction of energy content. In those studies, adult male rats were typically exposed to yogurt sweetened with glucose, which contained higher calories, or yogurt sweetened with low-calorie sweeteners. And reducing the correlation between sweet taste and the caloric content of food using artificial sweeteners in rats resulted in physiological dysregulation, like increase in caloric intake, increase in body weight, increase in adiposity, a decrease in glucose tolerance, and in energy metabolism. So it seems that altering the fidelity of the sweet intensity as a predictor of incoming calories creates a problem. We know that there is an association between taste and calorie content, and we can resonate with the well-known classical conditioning theory of Pavlov, that salivation occurs in the presence of food, while it usually does not happen when a bell rings. When adding this new cue, so the bell that is ringing, with the other cue, which is the food presentation, we will learn to associate the food with the sound of the bell and cause salivation. So after repeated learning, the sound of the bell alone will cause salivation without the food even being present. So the brain is constantly trying to predict consequences and anticipate response based on different cues. So usually, sweetness is an indicator of calorie content. And generally, the sweeter the taste, the higher its energy density. And since it learned to match the sweetness cues with the calories that it comes with. So the introduction of low-calorie sweeteners can cause a mismatch between the perceived sweet intensity and the energy level that is present, which can disrupt the body's response. So recent data from neuroimaging studies that combine fMRI with indirect calorimetry showed that sweet taste has an importance in regulating metabolism of glucose. So the group of Dana Small, they showed that sweet taste is important to regulate the generation of metabolic signals, and how it is really the optimal match between the energy content and the perceived sweetness that regulate the optimal metabolic response to the load and its rewarding values. So to explain more, in this study, to provide calories, they used maltodextrin, and to provide sweetness, they used sucralose. They also made sure to include people who were not able to detect the taste of maltodextrin in the drinks. So they created different beverages with innovative flavors, and sometimes they were new to all the participants. So the beverages varied in the amount of maltodextrin, ranging from 0 to 150 calories, but they all had the same sweetness level. So they use a concentration of sucralose that mimics the sweetness of a 75-calorie beverage if it was sweetened with sucrose. So this in the middle is the perfect match. So this had 75 calories with a sweetness level that was matching the 75 calories. And so people consumed those beverages during 2 days each to learn about flavor and post-oral effect. So as you can see here, the neural response in nucleus accumbens, which is an important part of our pleasure and reward system, was greater for the beverage with the optimal match between both the sweetness and the caloric content compared to the other beverages. So in the study, they also looked at the effect of matching or mismatching sweet taste and carbohydrates on energy expenditure. So they did it by giving people different solutions with either 150 or 75 calories and with match or unmatched sweetness. So for example, here, if we see 75-75, it represents a beverage with 75 calories and a matched amount of sweetness, while 75-150 represents a beverage that has 75 calories, but higher sweetness, so sweetness that is equivalent to a drink with 150 calories. Same applies for here. So in green, we will see the beverages that match calories and sweetness levels. And in orange, we will see the unmatched drinks or beverages. So they also used indirect calorimetry to measure the diet-induced thermogenesis. So this is the increase in energy expenditure that is due to the cost of processing food for use or for storage. So they examined the changes in the resting energy expenditure following consumptions of the beverages. And we can see how the higher resting energy expenditure, so here is the difference, we can see how the higher one occurs when the beverage is matched with the calories and the sweetness. So people are able to metabolize better beverages with matched calories and sweetness. Furthermore, there is an association between the metabolic activity and the brain response. So they observed that the higher metabolic activity here occurred with the higher brain response and nucleus accumbens. So the best way the body metabolizes glucose and the best rewarding value of that sweetness is when there is a match between the level of sweet intensity that is given with the level of calories. So this study shows an important and unanticipated role for sweet taste perception in energy metabolism. So perhaps looking at the phenotype of what happens in the mouth can explain a little bit how sensitive we are or not to the metabolism of glucose in the gut. So we discussed earlier how drinking sucralose or water 10 minutes before an oral glucose tolerance test resulted in different metabolic responses. So if you recall, in this group of people that we studied, we saw how they respond to an oral glucose tolerance test when they drink sucralose or water 10 minutes before drinking glucose. So sucralose increases the glucose peak when compared to drinking water. We will focus on the difference between the two, so the difference between the glucose peaks here. So we also evaluated them on their sensitivity to detect sucralose. So what was the smallest concentration that they require in order to perceive a taste that is different than water in their mouth? So we can see here on the x-axis that on the left side are the people who are the most sensitive to detect sucralose. So they require a smaller concentration compared to those on the right. The difference in glucose peak is represented on the y-axis. So you can see that this association is showing us that the bigger the difference in the glucose peak between the water and the sucralose, the bigger the metabolic effect of swallowing sucralose is observed, which means that the people that are most sensitive to, which means that it occurs in the people who are most sensitive to detect sucralose in the mouth. So in other words, the more sensitive the sweet taste receptors are to perceive sucralose in the mouth, the greater the increase in glucose peak caused by drinking sucralose before the OGTT. So these data also add to the evidence of a link between sweet taste perception and glucose metabolism. So another study that looked at sweet taste perception and glucose metabolism analyzed taste detection in people with type 2 diabetes and their relatives. So people with diabetes, type 2 diabetes, were found to have a specific impairment in, oh, sorry, I just got a message that I may have difficulties in the connection. Can you hear me well?
Yeah, I can hear you perfectly. It might have been just for a few seconds.
Oh, okay, great. So people with diabetes were found to have a specific impairment in glucose taste detection. And in this study, they raised the hypothesis that taste alteration can also reflect a defect in glucose sensitivity, including glucose receptor cells. So it was assumed that non-diabetics who are related to diabetics could carry a genetic predisposition for diabetes and would also express taste alteration as a genetic marker. So in the study, non-diabetics with their first-degree relatives who have diabetes were included and matched with controls. Participants with diabetes as well as their relatives showed a higher mean threshold than their respective controls, which means that people with diabetes and those related to them showed a decreased sensitivity to glucose compared to their respective controls. And this was not the case in people with juvenile onset diabetes. So the findings show relative alteration in taste detection that is linked to type 2 diabetes, but not type 1, and that it could be transmitted through genes. Now, this is there is more evidence that there is a link between sweet taste in the gut and glucose metabolism. Putting all this background together, the overall goal of my current research study is to explore the sweet taste signaling on glucose metabolism. So the current study is a logical continuation of previous studies. In all of our studies, we looked at people who were non-regular consumers of low-calorie sweeteners. And we know that there are studies looking at the fMRI and the brain and how the brain responded differently to sweet taste in people who are regular drinkers of diet soda compared to non-regular drinkers. So there is some questions that arise, like, is there a difference in metabolic response in people who regularly consume low-calorie sweeteners compared to those who rarely do so? And is there any difference in the sweet taste response in the people who regularly consume low-calorie sweeteners compared to those who rarely do so? So our goal is to determine the role of sweet taste signaling in the regulation of postprandial glucose metabolism, and to determine how acute and chronic consumption of low-calorie sweeteners may affect sweet taste perception and glucose metabolism in people with obesity. So we decided to continue selecting people with obesity since it is the group known to be consuming most low-calorie sweeteners and who receives the most advice to consume them. And it's also the group where we replicated the results and where we saw the changes in insulin in the previous studies. So the first aim is to determine an independent and combined contribution of oral and post-oral sweet taste signaling to the metabolic response to an oral glucose tolerance test in habitual and non-habitual low-calorie sweetener consumers. So we are comparing glucose and hormonal response to regular OGTT when the T1R-mediated sweetness signaling is pharmacologically inhibited by lactosol, both in the mouth and in the gut, or only in the gut. So our hypotheses are in non-habitual consumers. When comparing an oral glucose tolerance test with lactosol versus an oral glucose tolerance test without lactosol, the expected outcomes are that in those non-habitual consumers, inhibiting the sweet taste in both mouth and the gut will increase insulin response without decreasing plasma glucose, so reducing insulin sensitivity. Now, in habitual consumers, also when they will have an oral glucose tolerance test with lactosol versus no lactosol, the inhibition of sweetness will not affect insulin responses. And all three OGTT will reveal more insulin resistance compared with the non-habitual consumers. Also, in the non-habitual consumers, such reduction that was observed previously in insulin sensitivity when lactosol was administered will be ameliorated by sham feeding sucralose before the OGTT. So we hypothesize that we will have an improved insulin sensitivity when sham feeding sucralose compared to when lactosol is administered in those non-habitual consumers. As for the habitual consumers, sham feeding sucralose before the oral glucose tolerance test, we expect no increase in insulin sensitivity. Another aim that we have for our study, so we will explore association between subject sensory data and their metabolic response to a glucose load in the presence or absence of sweet taste. So first, we will determine sweet taste sensitivity and sweet preference in habitual and non-habitual low-calorie consumers. And then we will explore whether individual differences in the sensitivity to sweet taste correlates with the effect of sweet taste on glucose homeostasis. So the hypothesis is that people who are heavy users are less sensitive to detect sweetness. And for our second hypothesis, so it's still exploratory, but we expect that there will be a stronger relationship between metabolism and sweet taste sensitivity in those who are non-habitual users compared to those who are habitual users. So stay tuned for the results. Hopefully they'll be coming in the next 2 years. In the meantime, some studies have looked at the effect of replacing soda with water in individuals who usually consume diet sodas. So the study was done first in women who were overweight or had obesity, and then it was replicated in women who had type 2 diabetes. And they wanted to see if diet soda, what would happen if those regular consumers of diet soda replaced the drinks with water. So what they observed is that when replacing diet soda with water, there was a greater decrease in weight, so better weight loss, better fasting plasma insulin, better insulin resistance, and better 2-hour postprandial glucose results. So a lot of studies are showing that diet sodas might be better than sugar-sweetened beverages, but water is the beverage to go compared to diet sodas. So replacing diet sodas with water leads to even better results. So coming back to the take-home message that Dr. Pepino said earlier, regardless of the calories, let's use sweetness as a treat and not as a mean to a healthier lifestyle. We would like to acknowledge the American Diabetes Association for funding this research, and the USDA, National Institute of Food and Agriculture, for the fellowship they're providing for my studies. And we would like to thank everybody for attending today's webinar. And I think we're ready for questions.
Great, thank you, Clara. I'm just going to take the screen back here. We have a few questions, but as we go along, if anybody has any others, please feel free to type those in. Okay, I'm going to take the screen back later. Okay, so one question about, Clara, your work, since you were the last person to go. When you were talking about, I guess it's both of you, when you were talking about determining who is a habitual user, how do you define that? For example, are they users of low-calorie sweeteners in general, or of sucralose, or is it that they use them on some kind of regular basis?
So we do have a questionnaire that we ask, that we provide for the participants whenever they come for a screening visit. And we do ask about a range of products that they consume. And usually, non-consumer would be defined as somebody who consumes less than one diet soda or the equivalent per week. And a habitual users will be somebody who consumes more than one diet sodas or the equivalent per week. And if they fall in the middle, they wouldn't be in any of those two categories.
Got it. Great. Perfect. So one question about the Dana Small paper, the recent one, since you spent a lot of time reviewing that one. So there's just a question here about the treatments between the groups. So one group received sucrose and sucralose, and then in the other group, they received maltodextrin. So the question is about the glycemic index difference between those two, sucrose and maltodextrin. And it might be quite different. So would that be any reason to explain the results that were found?
I'm not sure I completely understand the question. So in all of the studies, they use maltodextrin and not sucrose.
Okay, might have been that.
Just to explain it a little more, so they use maltodextrin to provide the calories, but they made sure that people could not detect maltodextrin in the drink. And the sweetness was coming only from sucralose. But the amount of sweetness that was coming from sucralose was matching a drink that would normally contain sucrose. That's why sucrose was mentioned. But sucrose was not used in any of the drinks. It was only maltodextrin.
Okay, and Dr. Pepina, did you have anything to add?
It was to add that clarification that perhaps the confusion came with mentioning that the 75, that the level of sweetness used in all the solutions, as Clara mentioned, was like if you had a beverage prepared with 75 calories with sucrose, how sweet it would be, like around 5% sucrose. So all the beverage with a different amount of maltodextrin had the same level of sucralose sweetness as if it was that sugary concentration that Clara mentioned. I think that's when the confusion came.
Okay, perfect. And in some of the differences between sucralose and water, so for example, there was a 16% difference in insulin excursions. Can you speak to the clinical relevance of those differences? Do we know anything about that, or is that more work to be done?
It's the easy question that I can always answer. There is more work to be done. I just don't know how don't know as a clinician. I'm not a doctor, and I don't know how a clinician will interpret that 16% difference in an insulin excursion. I think that any increase in insulin that doesn't come up with also a reduction in glucose response. So any increase in insulin resistance, I don't think is a good is a good sign for glucose homeostasis. I don't know. I cannot pinpoint on how good or how bad it is. I can only answer to the question of is it inert or it has an effect? Mm-hmm. And it has an effect. And again, is it clinically relevant? I think that we need more data, but I think that when interpreting what it does, I like to quote that this is what happened on only one event with one kind of diet soda. So we need more data to understand, does it replicate if you have 3 diet soda a day? Does it every time, does it get accumulated? Is the response worse, or does it adapt and it gets normal and does nothing? But even if it was just as any increase in 16% of every single time that you engage into that behavior, I think that we need to consider that these low-calorie sweeteners, these are things that we don't consume for 1 week or for 1 month, or it's not a medication of a few days. It's something that we engage in consuming for years. And so if you are adding something that it changed, if I will do like the parallel with, for example, a drug that it changed maybe 1 or 2 times your heartbeat, it increased your heartbeat for 1 or 2 times. I think that things that were used in a chronic way and for many, many years, they can all add up and maybe can explain some of the effects that we see only in studies that are prospective studies that takes a lot of time and many years to see what the metabolic effect could be.
Got it. Okay. So here's a question about the fact that in the marketplace, a lot of products contain both sweeteners with no calories and a sweetener with calories. And there are pathways for both of these on individual cells. So the question is, do you think these pathways interact at a cellular level? For example, is it possible there's a need to sort of prime the metabolic pathway when you activate the sweet receptor to elicit a normal response?
I am confusing the question on the part of that there are pathways for both on individual cells. There are also pathways for them in the same cell. So, for example, in the enteroendocrine cell in the intestine that now is called like the neuropod, we know that there are expressed sweet taste receptors and sodium glucose transporter 1, for example. And so we know that glucose, for example, can activate both the T1R3 and also the SGLT1. But for example, sucralose can only activate the T1R3 without the SGLT1. However, we also know that the activation of the sweet taste receptor can upregulate SGLT1. So I think that they can prime, they can activate both, although they can have similar and they can have different and shared mechanisms, sugars with low-calorie sweeteners. We also know that by activating the sweet taste receptor, they can also modify perhaps the expression level of glucose uptake. And so I think that there is a priming. They can both do something. For example, when you mix a product that has low-calorie sweeteners and carbs, can cause something perhaps different that you only drink a diet soda with no any other carb, with no food, with no other beverage. Maybe the effects are different, but we need to do the testing.
Okay, so yeah, the question was further clarified. So the question was really about within a cell. So could there be crosstalk between these two pathways?
In the same cell, a crosstalk? No, I'm not sorry, I'm not understanding. And also, are we talking on what happened at the level of priming, like by cephalic response, how you may respond differently to gut hits in the intestine and how to be handled in the gut? Or are we talking on priming within the same cell, like in the intestine?
I think the question is about the intestine.
Yes, so there is robust data in preclinical models that there is an interaction. The pathways interact at the cellular level. The sweet taste receptor interact with sodium glucose transporter, and so it can interact it can affect modified glucose uptake. We also know from data in humans that there is a dysregulation of the sweet taste receptors in the intestine that is associated with obesity, for example. And also that can explain differences in the rate of glucose absorption in the intestine. So they are talking the sweet taste receptor with the sodium glucose transporter in the enterocyte, enteroendocrine cells.
Okay, super, thank you. So you primarily studied sucralose, but given that different sweeteners, I don't know if you can speak to this, but different sweeteners are metabolized differently, would you predict different effects across all the sweeteners?
Clara, do you want to take that, or do you want?
I'll let you answer it.
I think that since we talk about potential mechanisms and we said they are not mutually exclusive, I think that there are some mechanisms in which definitely different low calorie sweeteners will have different effects because of the different metabolic fates. So for example, if you think on the microbiota and how they may affect microbiota, it may be different if one if the artificial sweetener make it all the way to the intestine in the colon, or if it's metabolized early on in the small intestine. They will have different effects in microbiota, perhaps. I will hypothesize. If it was for activation of even sweet taste receptors in the intestine, again, depending on the fate of the metabolism. For example, if you think on aspartame, it is metabolized into amino acids and get absorbed. It will not maybe make it all the way to activate some of the sweet taste receptors. However, if there is an effect also, if we incorporate the effect that it can go through cephalic responses, just activation of sweet taste and how that primer of the sweet taste in the brain activates in a different way, primes the gut to handle that glucose differently, then I will argue that then low-calorie sweeteners in general, anything that activates the sweet taste through the brain will have a similar effect. So then at the end, it's a result of how many of these pathways are modified and what will be the net results of the different mechanisms. Then different low-calorie sweeteners will have different effects.
Okay. Thank you. And I think we just have one more question. Both of you can answer, but it might be targeted to Claire. So Claire, you have studied public health nutrition. So Dr. Pepino talked a lot about the innate drive for sweet. So if you were thinking of translating some of this information for the general public, realizing there's a lot more to learn, and the drive for sweet, how might you present, let's say, recommendations to the general public?
So it's like the last point that we touched on. So of course, a lot of people are attracted to sweet, and we always are looking for more sweet products. And the problem is that a lot of people are recommending low-calorie sweetened products compared to sugar-sweetened products, which might be better, but we still don't know to what extent they might affect us. So if we wanted to give some recommendations, is just be careful of how much you consume. So everything is moderation is, of course, better. So just be careful of how much you consume, because we still don't know to what extent those low-calorie sweeteners might have an effect in the body. Oops, I'm seeing other questions. So I don't know if Dr. Pepino, you would like to add to this?
I think it's realizing that biologically we're vulnerable to go for sweetness, and it's a source of pleasure. And so we're going to be trying to consume more and more. So trying to understand that maybe our world today is overly sweetened, and not, maybe to learn to like other things, to learn our beverages that doesn't have to be sweet. Your coffee doesn't have to be sweet. Your tea doesn't have to be sweet. Start to learn different things maybe don't have to be sweet. Trying to learn on enjoying the sweetness of a fruit. In particular for children, if we start offering to children a lot of the different low-calorie sweeteners, of course, with the exception of children with diabetes. If you have a kid with diabetes, we know for sure it's much healthier to get them in one event a low-calorie diet soda than a regular soda, not to overload their system with a sugary load. But just even a kid without diabetes, just to offer them a diet rich in sweetness, because we give them low-calorie sweetness because they have no calories, it may not do any good for them to learn, for allowing them to learn pleasure and learning to accept and start liking vegetables or even other fruits that don't take as sweet as other things that we offer them intensely sweet. So I think we just need to understand that sweet seems to have a very important role as a signal, like the brain is constantly trying to predict how to better equip their response to handle the consequences of what we do when we are eating. And sweet seems to be a very good predictor in evolution, has been a very good predictor. And so now trying to work on that disrupting that signal fidelity of sweetness in how we handle, for example, carbs, it really may not be a good idea. So maybe just when you drink something sweet, go with the calories and take it in moderation.
And I would also like to add to this that maybe it's not realistic for someone who is used to a lot of sweet products and an extreme sweetness level in the product to completely stop. And that's not what we're saying, but it can be done by adapting slowly and by learning that, okay, maybe I will reduce a little bit tomorrow the sweetness. And with time, we can get to a lower liking for products with less sweetness in them. So it's not a sudden change in something that's super sweet to something that's not sweet. It has to be done slowly, and it has to be learned slowly.
But actually, I want to add, it has to be, more research needs to be done. We need to really understand how we can help them. And so we can say, you know, how to win or how not to win, but I think we really need more research, more data to guide general public. But I feel like we are safe by saying, like all the now, most of the governmental and non-governmental health agencies, to replace your sugary beverage, in particular for water.
Great, thank you. And this kind of relates to the next question. We got one more. So this is interesting. Apparently, in some Middle East countries, it's a practice to provide purified sugars as the first treat for newborns. And the idea is this helps to regulate glucose and energy metabolism. So do you have any thoughts on this practice?
That's very interesting. I didn't know about that. They were doing this as a tradition for priming or improving glucose metabolism. I am very familiar with the tradition on the Quran to receive the newborn with a chew date, like mothers, parents, they would chew a date, and it's even in the Quran. So to give them the welcoming and give them pleasure in this world. And I always relate it to the fact that we know that is such an effective painkiller, such an effective release of, there is an effective release of endorphins in babies. And so I was feeling like, you know, calming down and feeling them good. And I don't know, I really don't know about data that it will show that if you start giving them babies this sugar, like refined sugars, will improve their microbiota or their metabolism for carbs. I will be, to me, hard to believe. I think that there's nothing better for a baby than colostrum of the mom as the first things to receive. But I'm really not familiar with that literature.
Okay, thank you. Well, we all learned something there for sure. So I think we have addressed all of the incoming questions, so we can probably wrap up for today. We just wanted to thank Dr. Pepino and Clara for sharing your work with us, and remind everybody to please visit our website and register for the next webinar in this series. So that's going to be next Friday at the same time. And that's focusing on omega-3 endocannabinoids and their role in neuromodulation. So we hope you'll join us then, and everybody have a great afternoon.
Thank you. Thank you, everybody, for coming to our webinar, and thank you again for this invitation.
Thank you.
Thank you.